Thanks everyone for coming. Thank you everyone for coming to the IPM speaker series this month. And I'm pleased to introduce our speaker, which is Dr. Matthew Might. Matt is the director of the Hugh Kaul Precision Medicine Institute at the University of Alabama at Birmingham. He's the Hugh Kaul Endowed Chair of Personalized Medicine, a professor of internal medicine, and a professor of computer science. His research focuses on precision prevention, diagnosis, and therapeutics using genomics, model screens, drugs screens, and artificial intelligence. A principal theme of his research is the use of computer and data science to enhance clinical and academic medicine. Matt received his PhD in computer science from Georgia Tech and spent nearly a decade as a professor of computer science at the University of Utah. In 2015, he joined the faculty of biomedical informatics at Harvard Medical School as a visiting professor and as a senior lecturer. At Harvard, he focused on rare disease discovery and diagnosis and development of personalized therapeutics. From 2016 to 2018, Matt served as a strategist in the executive office of the president of the White House. At the White House, he worked primarily on President Obama's precision medicine initiative with both the NIH and the Department of the VA. In his talk, Matt's going to focus on the power and potential of precision medicine, but note and try and deal with the challenges in scaling this kind of ultra-individualized treatment. I think this is something we all think about a lot, and I think will be highly relevant. Matt's going to sketch an algorithm for precision medicine to illustrate a general roadmap for patients while also drawing attention to the specific bottlenecks and the opportunities within the process. And the title is Precision Medicine, Bringing N of One to Everyone. Matt.
All right, thank you. It's great to be here. And yes, I will try within this session to sort of sketch the contours of this algorithm that we've been sort of building as we, as we need it to try to treat patients that reach out at ever increasing scales. So I do need to start with a disclaimer. Disclaimer being that while there's lots of medical advice in this talk, I'm not qualified to give any of it. I don't have any formal medical training. I really am a computer scientist that has stumbled into precision medicine. And I'll end this talk actually with how it is that a computer scientist has ended up exactly in this position. And so a bit of my personal journey, which also I think colors my interpretation and my approach to precision medicine. And not only should you not take the medical advice in this talk without consulting your physician, I do want to even emphasize that all of the medical advice in this talk was actually generated by a computer. And part of that is because I think to achieve the scale that we really need, much of the heavy lifting will have to be done by machines. What's ironic is in just the past couple of months, things have changed quite a bit in terms of the believability of this claim. Pre-ChatGPT, it might have seemed fantastical that we'd have computers capable of helping us to scale this process. And now people go, well, maybe it is doable. So we'll even address some of that because a fair chunk of this is actually going to be about the evolution of artificial intelligence within precision medicine. So all this is to say I'm a computer scientist, and for a large fraction of my academic career, I focused on things like cybersecurity and high performance computing. I did do a little bit of medical devices early on as an assistant professor. I was helping out to verify the safety of these MR-guided high-frequency ultrasound machines for tumor ablation, but I wasn't doing the medical part. I was just making sure this machine didn't fry people alive on the inside. Most of what I did really was sort of fundamental algorithms and data structures, really hardcore computer science, a lot of which had implications for artificial intelligence and still does. And now I've had this career pivot. So I want to get three things across today. I want to define, how we see our precision medicine. I want to give a sense of what it looks like in practice in the context of individual patients. And then, back into why I'm doing this, which explains sort of my personal motivations, but also, why am I taking this very computational approach in many cases? So what is precision medicine? If you go back to the beginnings, the first definition of it is in this National Academies report from 2011. And in this, there's a glossary. And in that glossary, you find almost an essay-length definition of the term, comparing it, contrasting with personalized medicine. And really, they end up saying that, functionally, they end up being the same thing. But here's what it means. If you are tailoring medical treatment to the individual characteristics of each patient, then you're doing precision medicine. That's fundamentally what this is. And there's been a lot of different competing definitions for it that have emerged since then. And I think I've been able to pull all of them back together and be faithful to that original definition by saying that precision medicine is really about optimizing health with data. And by that, I mean, it's by optimizing, I mean, it's a process, and this is a process that has inputs and outputs. The input to this process, which, I'm already alluding to the fact that this is an algorithm, the process is an algorithm. The algorithm takes an input, and the input in this case is all of the data you can get on a patient. And the first piece of data that I think got people excited about precision medicine was the clinical accessibility of the human genome. So once this became a thing that clinicians could engage with, there was, I think, right away a recognition of, given its size and complexity, there was no choice but to invoke computation to help with this. But there are other sources of data that can be used as part of this optimization process too. There's all the data, longitudinal data, sitting in the electronic health record. And it's not like this had been fully exploited either. It still isn't fully exploited. So this can be part of this optimization process. And then I always like to point out that there's data outside of the patient that counts too. There's all the knowledge and data out there in published papers and biomedical data sets and databases. All that needs to be brought to bear on this optimization problem too. And I'll talk about techniques for doing this. But fundamentally, if you want to imagine this at a very high level, what we're trying to do in precision medicine is modulate things like lifestyle and treatment in order to arrive at the optimal health for a patient. So you can sort of split this process into two parts. And in practice, it really is kind of split into two parts. There's essentially a precision diagnostics phase, and there's a precision therapeutics phase. Now, in the precision diagnostics phase, you're searching for molecular diagnosis. I think this has become most salient in the context of rare genetic disease, where things like genomic sequencing allow you to jump almost directly to the root molecular cause of the disease. The only reason I say this is that, it's not inconceivable that we're going to do this at scale for more common diseases too, where we can really individualize, and identify the root molecular drivers of disease in many common disorders, which could vary for the same disease from patient to patient. There's technologies like transcriptomics and metabolomics, all of which may do for common disease what genomics has done for rare disease, and to a similar extent for cancer. So I'm not going to talk a whole lot about the precision diagnostics phase, because I think this is something that's more well understood. I've focused most of my energy on the precision therapeutics phase. Once you have this root molecular cause, what do you do about it? How do you match that cause over to a corresponding treatment? And so this is really the question I want to answer. How do you do precision therapeutics? And at a high level, you're going to need some kind of process. You're going to need an algorithm, like what I've just sketched out, that takes all this data and this information and gives you back potential treatments. So, and once again, sort of drilling down one level of resolution, there's really two branches at the top of the algorithm. And by the way, you can take both of them. You don't have to take just one or the other. There's what we think of as the rational approach, or the rational side of the algorithm, and there's an empirical approach too. But in the rational side, you're trying to rationally invert the harm done by the core mechanism involved in the disease. Now, again, if you're thinking in terms of rare genetic diseases, sequencing might jump you straight to the gene driving the mechanism. But what that will not often do these days is tell you how. So you might be told this genotype is pathogenic, but the diagnostic report might not tell you what exactly is happening. So, for example, it won't tell you, is this a gain of function or a loss of function? And this is actually important if you want to treat it. If you're trying to rationally invert this mechanism, you have to know, is it overactive? Is it underactive? Is it absent? Is it toxic? Because there's a different strategy in each of these directions. And so as the algorithm gets narrower and narrower, as you drill down, we have a different strategy for activation, for inhibition, for compensation, and for elimination. And this approach really contrasts with other ways of doing it. And the other principal approach we take is what we call the empirical approach. So in this, you may not even know what the mechanism of harm is really doing. You have to have your hands on a mechanism because you need that mechanism to build a model. And it could be from cells, it could be a fly, or it could be some animal. And then the idea is to just test things on that model. So for example, an empirical approach would be to do some kind of drug screen on a precision disease model. So here you start with your patient, you extract from the patient a disease model. It could be cells from the patient. It could be cells you created based on information from the patient. And it could even be an animal model. And oftentimes for us, it is an animal model. The whole point is you have to have some model that you can study, that you can phenotype. And once you have that phenotype, once you have that signature of disease in the model, you can then convert this into a drug screen by combining the model with as many drugs as you can to see if any of them exhibit some sort of rescue effect. And in theory, and in some cases in practice, you can then test all approved drugs, and you can even throw in lots of natural products. So these kinds of screens will vary between 1,500 and 2,500 compounds. So they're in the grand scheme of things, these are not considered sort of classically high throughput because those can be into the millions of compounds, but it's still enough that you want to make it a very scalable readout to do something like this. Now, if you do this, the hope is that something actually rescues this model. And then you also hope that whatever you found scales back to the level of the patient. And what can happen here, just I'll tell you from experience, is it could be that you found maybe you find an approved drug, and it rescues in the model, but at concentrations that would correspond to impractical or infeasible doses in humans. That happens a fair amount. And at that point, it becomes a medicinal chemistry problem where, can you manipulate the potency or turn down the toxicity so that you can achieve the right concentrations in a human body? So what does this look like in practice? I'll describe to you what we do at the Precision Medicine Institute at UAB. And we're really guided by a core mission here, which is to do science in the service of patients. And I will describe this through a series of examples now that will allow us to back into some more general methods. I have to give credit, of course, to the amazing team we have. We have Madeline Eckenrode as our medical director, Tony Farguson is the deputy medical director, who also happens to be the chief medical officer at our local children's hospital. And that's sort of a sign of his commitment to bringing precision medicine to essentially everybody and to pediatrics within Alabama. Then we have a program coordinator, Alexandra Fuchsinska, who really makes everything happen. On top of this, there's also a very large team of undergraduates, and I have to give them credit because they do a lot of the heavy lifting. They take, a lot of the data that gets turned up by our computational tools. They do a lot of research. They compile drafts of reports that go to our medical directors. They then get checked over, edited, verified by them before getting passed on to the treating physician for a patient that has reached out. So these undergraduates end up interacting with, AI and computational tools. They end up interacting with physicians. They end up interacting with scientists, and they interact with patients themselves in many cases. To really coordinate, always finding the next step for every patient. So I'll share some stories, and I'll start with, one that's more common these days, which is precision oncology. This is Mickey's story, and I'm starting with this in part because this is actually a direction we want to go. We want to actually start changing what precision oncology means by using some of the techniques that we've developed for rare disease. Now, in Mickey's case, when he reached out, he was 56. He had prostate cancer. He'd been through 42 rounds of radiation treatments, 17 rounds of chemo. He was bedridden and near death. So, very hopeless. And his PSA was, essentially off the charts, where if this is normal and this is considered bad or worrisome, this was Mickey. So it was, I think almost 100, which is, again, just nightmarishly bad. Fortunately, our then at the time head of precision oncology, Eddie Yang, was able to step in, do tumor sequencing, and identify a driver mutation. Interestingly enough, the mutation happened to be one you typically see active in ovarian cancer, and he was able to use an ovarian cancer drug targeting that mutation for Mickey's prostate cancer. And remarkably, this actually worked. And so within 60 days, his PSA had plunged. And more importantly, his quality of life had really surged back, from essentially near death to out and about again. So that's Mickey's story. And I'll move on now to a non-genetic story. And I like to also include this one to start because it emphasizes the earlier point I was making about precision medicine involving not just the data about the patient, but all the data outside the patient too, and gives you a sense of what you can do if that's all you have. So in the case of Kelsey, she was 19 when her parents reached out, suffering from cyclic vomiting syndrome, frequently hospitalized, and very low quality of life. She was at 75 pounds, suffering dozens of vomiting episodes. And of course, as is often the case, when folks reach out to us, they say, we've tried everything. And this is practically the admission criteria for our research program. You have to say this. And sure enough, they tried a lot. I mean, everything was probably about 25 things. It's a lot of different things to try. But we actually read everything. And what that meant at the time, there was only 30 million published papers back then. And we found 64,000 on nausea. In between them, there's 4,000 papers on treating nausea. Drilling down even further, there was 347 distinct proposed treatment modalities. And this is where we get a sense of everything that a team has tried or everything that a team knows about versus everything that's really out there in the literature somewhere. And this is what everything looks like if you graph it. So you end up with a histogram ranked where treatments are ranked by the number of publications that support them. So if you look at, say, Zofran up at the top here, there's 169 papers that cite Zofran or mention Zofran as being an effective treatment for nausea. So that's a lot of papers. And as you go down this histogram, you start to find less common things. So eventually you get to the region where there's only 3 papers in support of the proposed treatment. And way down there, you find nasally inhaled isopropyl alcohol. And so literally just sniffing, rubbing alcohol during episodes of nausea. And there's, a small but meaningful amount of evidence that this is a reasonable thing to do. So graphically, there's this region here before the long tail. You can call that the known. This is everything that a team knows about or a single physician knows about. Then there's a big long tail, this unknown known, all the things that, technically our species knows, but may have entirely forgotten about. And, when I say we read everything, what I really mean is not we, but an AI. So natural language processing. And so we've been doing this now for about 6 years, so now everybody's excited about what ChatGPT can do. And of course they should be excited. It's genuinely impressive. And we're already leveraging its natural language processing capabilities to go even further. But we've known for quite some time the power inherent in this approach, and we've been using it this whole time to help patients. So, by essentially condensing all the literature into a single queryable database, we were able to send them back this evidence saying, have you considered this? So we sent them over. They said, well, we've never seen this, but we can try it. And it worked. Literally within the same day, we heard, hey, she is able to now abort every episode of nausea with this. So if you fast forward 8 months, things are still going well, in fact. So well, in fact, that not only is she no longer vomiting, but she's up to 125 pounds, and she is married. So the high school sweetheart who had been visiting her essentially every day in the hospital proposed, and they got married and moved to California. So I like to start here because there may be some genetic cause, there may be some molecular cause here, but in some cases, just better access, structured access to what's already known can provide clues about what to do. So the question, of course, is, can we do more? And then once you've digested the literature into a database, yes, you can do a lot more. So we have a tool. It's called Medicanron. Will Bird is in charge of this project. It's funded by NCATS, and it's part of this broader biomedical data translator program. And what it's really doing is it's bringing the full power, the full weight of logical reasoning to biomedical knowledge. So right here is one important differentiator between a tool like Medicanron and off-the-shelf ChatGPT. So ChatGPT, and I've been playing with this new version of a GPT-4, it doesn't use sort of pure logic. It uses logic-like reasoning, and so sometimes it makes inferential errors. But we're very strict with Medi-Cameron. We don't allow it to exceed the bounds of logic. And so it can justify every inference it makes for you. It can cite all of its evidence. And I won't get into it in this talk, but there are ways that this actually plays very nicely in an interactive fashion with tools like ChatGPT. So what we have in Medi-Cameron essentially is a tool that combines old school logic with high-speed automated reasoning to achieve superhuman-like deduction with clinical insight, or so the hope goes on this last one. And I can tell you what an AI brain looks like, at least in our version of AI. And it looks quite different than the artificial neural networks of ChatGPT, because this, when you peer at the innards of this AI brain, you can actually understand what's going on. Inside, if you opened it up, you'd see a collection of these nodes or circles, each of which is labeled by a noun, a concept from biomedical research or medical practice. And between these nodes, you'll find arrows, edges, and they're labeled by predicates. So every time you see an arrow moving from one circle, essentially one noun, to another noun, you have an encoding of a fact, a subject, a verb, and an object. And so this virtual brain is encoding sentences. Sentences are essentially the atomic unit of knowledge, if you want to think about it in a mathematical sense. So once you have them represented like this, you can do interesting inferences on top of this. So here you have an encoding of the sentence, previsite inhibits engase. And once you know that there's a relationship between two concepts like red and blue, you can use that to deduce or hypothesize other possible facts about related concepts. So for example, if blue increases red and red decreases green, you might infer that by increasing a decreaser, blue, logically speaking, should be a decreaser of green. So that's an example of an inference that you can make in a brain like this. And so in some sense, the brain inside Medi-Cameron is just it's this soup of nouns stitched together with verbs to form a database of sentences. That's what it looks like. And we can query this brain. We can ask it questions. We can say, okay, what are potential treatments for Y? Or what are potential inhibitors or activators for X? And an example I'll give you right now is a disorder called MAPK8IP3. In fact, I should offer a disclaimer right up front. There is more than one MAPK8IP3 disorder. So if you're dealing with patients that have this disease, it's important to be very precise about the specific variant itself and understand what is really driving the disease in their particular case. In this first case here, we actually were put in touch with a 5-year-old boy through the Undiagnosed Diseases Network, and he couldn't walk or stand. And his MAPK8IP3 we deemed to be underactive, effectively. And in this case, it was much easier to make this determination. It was a very clear loss of functional allele. So no real doubt that this is what we're dealing with in this case. So essentially a haploinsufficiency. That's why I said you have to be careful. It's not always the case that you have loss of function pathogenic alleles. You can sometimes have some that appear to be both loss of function and dominant negative at the same time. And so the query in this case was pretty straightforward. It's what activates this gene. And it came back with an answer, almost immediately, literally less than a second. It said, it looks like I have a fact somewhere in my head, or a series of facts that allow me to infer that high-dose retinoids should potently upregulate this particular target. So nothing happened we actually took all this information, got it back to the treating physician, and nothing happened right away, actually. So it was a bit anticlimactic. But the mother stayed the course in terms of using the advice given to her by her son's pediatrician. And sure enough, 6 months in a disease where this child had yet to stand and certainly not even walked, he was starting to stand up. So actually standing briefly under his own power, and even taking very crude steps. So this video here illustrates that. So very excited video from mom when we got this. And also just to give her some credit, at the same time that they were doing the recommended treatment, she also kept pushing him to reach whatever his potential was in physical therapy. And it really paid off because, within 12 months of that, we got another video. So a year later, um, not only is he standing up, but he's standing up under his own power. And he can even drop down, he can stand back up. You'll see him go all the way to the floor in just a second. And so again, these videos, they're only 12 months apart. And so we're going from 5.5 to 6.5. And it's a pretty dramatic difference. And especially when you consider that, he was completely non-ambulatory 18 months prior. So significant progress. Now, there's been a lot of research on MAPK IP3 since then, and we do have a much better understanding of the mechanisms by which retinoids could be working in this case. And interestingly enough, it appears that they're probably only going to be effective during development. It looks like they are, in fact, impacting some of the issues with development, specifically in the brain. And so it's probably really important to have this during the developmental window. That wouldn't be very effective later on in life from everything we've been learning. And we've kept going. One of my faculty, Cami Crowder, made a gene-edited zebrafish model of the disorder. And over the summer, was able to find rescues in this model by testing drugs on it, predicted by Medicameron, one of which was amantadine. So amantadine seems to rescue the zebrafish model. And this, in turn, seems to be making a difference in the patients as well. So this approach, using precision animal models is so important to us that we actually now have a Center for Precision Animal Modeling at UAB. I'm the co-director of it along with Brad Yoder. It's been generously funded by ORIP, and it allows us, and this is potentially of importance to folks on your side, that to build this precision disease models, for patients that reach out, for physicians that reach out. And once we have the model, we can do things like test drugs on them. So the next case I'll show you is an autism. In this case, it's genetic autism caused by a haploinsufficiency in ADNP. And what happened here, before Medicameron even existed, this mother who I'd known for years, and she reached out, she's the head of the ADNP Foundation, and she said, I heard you're building this new AI tool. Is there any way that you might be able to see if there's something out there for my son or for ADNP more generally? And so by February 2019, we actually had the tool up and running to a point where we could try it out and ask this question. We asked, hey, what upregulates ADNP? And it came back with an answer, less than a second, low dose ketamine. So there's strong evidence that we can pull together from the literature that this could be a potent way to upregulate ADNP. By February 2020, so we took all this information, we passed it back to the foundation, sent it to their chief medical officer specifically, and said, hey, here's the research that's out there. This looks real to us. He wrote back literally the same day and said, oh my gosh, this looks very, very real. The foundation then started doing everything that was necessary to basically get through, the preclinical stage of a clinical trial. Say, let's go ahead and do this. Now, fortunately, ketamine's been well studied in humans, so there's plenty of data to repurpose for all of this. And they actually had approval for a trial by February 2020. This was the announcement on the patient blog saying, hey, we've got the go ahead. Of course, COVID hit, but it didn't deter them. They actually launched by September 2020. And so this is an Instagram post that they put out as the trial took off. And so very exciting that they actually pulled it off even during COVID. By one summer later, they'd finished the trial. And in fact, I think there were 14 different endpoints measured in this trial. One of the really interesting ones from my perspective was the original hypothesis. Was ADNP expression doubled? And sure enough, it looks like it was. But perhaps, in fact, actually, definitely more importantly, there was also significant clinical improvement across the board and almost everything that was measured. And so that's really what we wanted to see. If you're interested in more of the details, it's been written up in Stat. It's from a couple of years ago that, and it really just goes into detail about what the difference that's been made in the lives of the patient as a consequence of this, and how we got to this point using artificial intelligence. And if you're interested in the more low-level details, I and others published this back in October, the details of the trial itself. So you're welcome to read that. Next case is actually also from the Undiagnosed Diseases Network. And here we have an overactive potassium channel. And you may have actually seen this case on Netflix. There's a show called Diagnosis, and this is one of the featured patients on that show. So she was the first patient in the world to be diagnosed with this particular gain of function in what they call the big K, or big potassium channel, KCNMA1. It was causing on the order of 150 cataplexic episodes per day. So quite severe in terms of impact on her. And initially, we ran this through Medicanron. This is one of the first queries we ever actually did on Medicanron. And it said, well, it looks like methamphetamine is a potent inhibitor of this. And sadly, we kind of chuckled and disregarded it at the time. And I wish we hadn't. I really wish we hadn't, because it was a real hit. And just because you wouldn't necessarily give methamphetamine doesn't mean you couldn't give a different amphetamine. And it should not have surprised us in the least that it was not the methyl group that was doing the work. It was the amphetamine doing the work when it came to this inhibitory effect. So this is the case. It really is, most amphetamines will have this effect. In fact, every amphetamine we know of so far seems to. And of course, many ADHD drugs are based on this backbone. And in particular, Lisdex amphetamine seems to work particularly well for this. So I was sent over to the physician for this girl, and mom called not long after that. She called me directly and said, "Hey, it worked. We've actually, completely stopped all these cataplexic episodes. I mean, literally down to zero." So that was quite striking. And now I think we know of at least 5 patients around the world, probably more at this point, that have seen the exact same effect when going on Lisdex amphetamine. Because this is a UDN case, there's been work inside NIH to look for funding that might be usable for a clinical trial to start to verify this. We're looking at some pretty novel trial designs too. So for example, given how these drugs work, we think it might be possible to do like a single day withdrawal trial. We're just looking at blood levels over the course of the day, plotted against the number of cataplexic episodes. It might allow us to make inferences about what's really going on here. So for the final story, I'll actually tell you my personal story and how it is that I ended up in precision medicine and why it's shaped my approach to it. So I'm in this because of my son Bertrand, who you see here. He was, I think, 1.5 years old in this picture. And he was in the early stages of a diagnostic odyssey. By 6 months old, it was clear something was wrong. Didn't have a name, but something was wrong. And we landed on a place that many patients do, this undiagnosed island. And it's when you're on this island, you're trapped there with a population of one patient, or really just one family. And you don't know why your child is suffering. And in Bertrand's case, he had seizures, he had a movement disorder, he had developmental delay, and he had a curious lack of tears. So it's not that he couldn't cry, he just couldn't make tears when he did, which in turn led to a host of problems with eye moisture, required this really strict lubrication regimen, and was a major impediment to his quality of life. I mean, there were times when he would have eye surgery to drain the pus from his cornea from the abrasion of his ultra dry eyes. So it's not that the seizures weren't bad. It's just that hour to hour in most days, it was his eyes that were the most difficult to manage. And unfortunately, they also gave us a life expectancy of 2 to 3 years. There were serious issues with his brain, his liver, and his heart, really all at the same time, and not a lot of hope. And of course, no answer at the same time. We got very lucky that there was a breakthrough during the middle of all of this. It was exome sequencing. I'd been trying to get him genome sequencing, but it was just outrageously expensive, and it wasn't a clinical thing. And overnight, the cost fell dramatically with exome analysis. Again, not something you can do clinically, but Duke University reached out and said, we think you might be a good candidate for, the first clinical trial of exome sequencing for these intractable diagnostic odysseys. And so we said yes and signed him up. Turns out he had 2 mutations in a gene called NGLY1, both very clear loss of function. So it looks like, again, total ablation of this gene. And, they were reasonably confident that this was the cause, when they reached back out to us. So that he was, in effect, at this point, the first NGLY1 deficiency patient in the entire world. It also, by extension, the only NGLY1 deficiency patient in the world at that point in time. And I knew you can't have just one disease versus or one family versus an entire disease. You have to find others to work together. Fortunately, I had access to a secret weapon as a computer scientist. I had Google. Now, obviously, you can't just go to Google and say find more NGLY1 patients. You've got to, in some way, trick Google into sending the patients to you when they start Googling stuff. So it's not quite that simple, but that's what I did. I wrote a blog post, and this is the blog post right here. And it was really designed to serve as a beacon to attract patients to us. So there's, a catchy title up here, some clickbait, and then 3,000 words beneath this to do essentially search engine optimization to draw people in. And it actually worked. So 2 weeks later, there were 2 more patients that had been identified through this blog post as NGLY1 patients. And so at this point, there was a community and a confirmation. There was, maybe a shadow of a doubt that maybe we'd gotten the right answer. And once we saw these other patients, there was no doubt at all that this was, in fact, the disease we were dealing with. Led to my collaboration with this glycobiologist, Hud Freeze, who was probably the only person in the world at the time qualified to explain what was happening to Bertrand on a molecular level. If you want to know more about the birth of this community in its earliest days, there's actually a really nice New Yorker article written about this whole process that describes it from Bertrand's diagnostic odyssey all the way through the very first meeting of these patients and even the birth of the Undiagnosed Diseases Network. So that's one thing that happened as a consequence of this diagnostic odyssey is I got involved with this Undiagnosed Diseases Network funded by the NIH. At the time, I was, saying initially like a patient advisor to it. Now I'm one of the principal investigators for the Coordinating Center, which is joint between UAB and Harvard. And finding more patients also enabled a natural history study. So folks like Bill Gall and Lynn Wolfe said, well, since you have patients, since this is a disorder of glycosylation, we're going to allow you to enroll them in this natural history study, which was amazing. So patients started going to NIH to be phenotyped at extreme depth and give us a better understanding of this disease. At the time, I was at the University of Utah. I ended up with a second professorship in pharmaceutical chemistry, because I really want to understand the process of developing therapeutics now. Previously, I was deep diving in genetics, and now I was getting into really personalized therapeutics. And through all this, enabled me to make a prediction. And I have to give credit to Lynn Wolfe at NIH for helping me with this. But I said, well, under the logic of this disorder, if NGLY1 is broken, what else is broken? So what's downstream of NGLY1? It's an enzyme, as it turns out. Enzymes have inputs and outputs. And if the enzyme goes away, the inputs should go up and the outputs should go down. And it turns out that one particular thing went down. And that was actually quite important. So I said, oh, he's missing something, under the logic of this disease. And what he's missing is an acetylglucosamine. And at the time, this was just a symbol to me, that I could type as a word, I could type into Google and say, what is this? And found out very quickly that whatever it was, you could buy it on Amazon. In fact, you could buy large quantities of it on Amazon. And that's exactly what I did. So I bought a bunch of this stuff on Amazon. I took it myself. And I woke up alive the very next day. And for me, that concluded FDA phase 1 safety testing. And then, I said, okay, I think I could imagine giving this to Bertrand now, because in my household, I am the FDA. And you'll find quickly that's how it works in every rare disease household. Now, it wasn't right away that I gave it to Bertrand. There was some contemplation involved around the ethics and the science of this. And in fact, it took, I think, 3 weeks to sort of reason my way to it. He accelerated the reasoning by going to the hospital and almost dying in the hospital. And I remember just thinking, if this was it, if this was what might have helped, and I never found out what it could do, I don't know how I live myself as a parent. And so once he was home, once he was stable, once he was sort of back to his baseline, I started putting this in his food. I got dosing guidelines actually from a study of the same compound in multiple sclerosis, where it seemed to be effective. And one morning, I think it was 3 days in, was feeding him his breakfast, or was late to get up feeding him his breakfast, and he was upset, he was crying, and he was actually making tears, which you can't not notice that. I mean, it is, yes, given the nature of his condition, it was such a striking feature to see all of a sudden, that I remember, pulled out my phone, and I did what every parent does in that situation when they see their child cry for the first time. I collected his tears, I packed them on dry ice, and I shipped him to that glycobiologist in California for analysis. And so, we ended up doing essentially proteomics on his first tears to try to distill what was going on. And as a result of this, these tears, eventually swelled ultimately to an ocean of science for the disorder as a whole. It really started to unlock some of what was going on in Bertrand. And people always say, well, it's great you can help humans, but we're biologists, and as biologists, what we really need to know is, can you do this in flies? And so I found Clement Chow, a human geneticist who specialized in Drosophila, and started playing with the fly version of this disease. So it turns out, if you take NGLY1 or the ortholog out of flies, they die. Only about 18% survive. But if you give them this compound, almost all of them survive. Think about 90% or so survive once you give them this. So if you want to know more about the details of this, it's all in this paper here. You can read about it. Clement did an amazing job, deep diving on this with transcriptomics and just really thoughtful analysis of what is happening in these flies, what's going on with Glucknack, again, just all the details are here if you really care. That's what ultimately led shortly thereafter, while I was on sabbatical, to this faculty appointment in biomedical informatics, and also to the founding of a company, Paranomics, to do personalized drug screens for epilepsy. Now, that company was acquired by Q State in 2018. And ironically enough, I haven't updated these slides yet. Q State was acquired by another company 2 days ago. So it's like these ever larger fish eating each other, I guess, at this point. And I was a board member at Q State until 2 days ago, and no longer. And then I got the opportunity of a lifetime. I got an invitation to meet with President Obama at the White House, and then an invitation to have a job at the White House. So I ended up working at the White House for about 3 years on the Precision Medicine Initiative. And I won't go into my White House stories here, except to say that, yes, I have some, and really enjoyed the opportunity to help lay what I hope becomes ultimately the scientific foundation for doing this kind of work for more people. Ultimately, after this, ended up at UAB. And of course, I kept trying to do everything I could for Bertrand. Got a grant to do real therapeutics development for NGLY1 deficiency, joint with Kubi and Yiling. And to do this, we made NGLY1 worms. And as part of that, we ended up discovering that they showed a pretty robust phenotype for NGLY1 deficiency, and allowed us to find other drug targets to go after. So I'm skipping over a lot of the details, but one of the drug targets that they clearly revealed, which actually lined up with what we were seeing with some work in Japan from Tadashi Suzuki in mice, was NGAs. So basically, we needed an NGAs inhibitor. We turned to molecular simulations to do this. And through these molecular simulations, testing around 200,000 compounds, we identified a number of potential inhibitors, 70 in total, 14 which were already FDA approved, and one which actually worked in the lab. And that one was Prevacid. So Prevacid is an NGAs inhibitor. It's also a proton pump inhibitor used to treat acid reflux, but there's no question that it is also an NGAs inhibitor. So if you want to know more about details about this, how this was done, it's all in this paper here, on how these inhibitors were identified. And over the course of his life, it did have a pretty big impact on Bertrand. Yeah, as more and more treatments stacked up. By the time he was really 7 years old, I would say he had no more clinical seizures for sure. No issues with his eyes whatsoever. But more importantly, he was just happy all day, every day. He was very happy. And I think that's, the metric that I really cared about for him, that I still care about for every patient we work with, is the one you measure in smiles per hour. He eventually improved to the point where he started to be qualified to use an eye gaze computer. So he was able to start building essentially his own language using eye gaze. And so, for example, when he would come home, he could say things like, hello, like Matt is my dad, which is Bertrand's way of saying, hi, Dad, welcome home. And by 2019, a clinical trial was actually funded for NGLI1 through a U54. I'm one of the PIs on this. Ava Moreva at Mayo is leading it. He was actually the first patient to go up and start with safety checks. Of course, then the pandemic hit. Trial was suspended for a while. And unfortunately, his health very slowly declined over the course of 2020. Ended up in the ICU multiple times, more than usual, in fact. And it prompted me to say, it's time for, getting out and enjoying life. Not knowing, if anything was imminent or if anything might happen, but I took him essentially on a sunrise tour. Took him to his favorite beaches, his favorite lakes. The lake house where I spent most of my childhood summers. Allowed him just, enjoy the world and enjoy his siblings, of course. And I'm glad I did, because here he was happy, healthy, smiling. And then 3 days later, he was gone in October of 2020. Literally the shortest hospital stay of his entire life. It was less than 48 hours. It was sepsis that progressed rapidly to septic shock. And still, beyond not being COVID-19, we don't know what drove it. It was just so quick. So all of this, of course, has given me a perspective on what really matters, not just in precision medicine, but I think in life in general. And I really do think it's caused me to double or triple down on the mission of science in the service of patients. And I'm very lucky that I'm able to do this. And I've collected a lot of resources in Bertrand's memory on a website form, bertrand.might.net, including a 90-some page written description of this algorithm for precision medicine that I've been stepping you through at a very high level here today. So if you want a resource that you or patients can work with, it's up there. And of course, the mission continues. This endowment established in his memory has funded a treatment for a novel genetic incarnation of acanthosis. So this patient was suffering these scaly growths all over her skin. Turns out it was due to a germline gain of function in EGFR. And using this endowment, we helped create a special topical cream version of that. So this is normally a cancer drug. We made a topical cream out of it. It took a year to figure out how to do that, but we did it. And it worked. It actually resolves these scaly growths for this patient. And there's actually now an IND approval for a larger N of 18 trial for NGLI1. So very excited about this in terms of continuing to push for therapeutics for his disorder. If you're interested in watching this process, you're certainly welcome to join us for case reviews Mondays at 11 Central, 12 Eastern. And you can zoom in and watch us do workups where we work through the specifics of individual cases, look at new cases, look at updates on old cases, and try to figure out what the next steps are for any individual patient. So with that, I thank you, and I'm happy to answer any questions that you might have.
Matt, thanks very much. That was a fantastic talk, very moving, and your perspective is obviously truly unique. I have a couple of questions. I think that's fascinating at the end, what you talk about how people can use your algorithm and then even listen into your diagnostic board. Could you explain that just a little bit quicker? Because there might be people, I'm sure there are people actually at our institution that would be interested in doing that. So if they use your algorithm, they would then have to then take that evidence to the IRB to suggest a new treatment for someone, new treatment paradigm?
Yeah, well, that's a good question, actually. So what if you find something? How do you actually take that back to the patient? So we thought initially, yes, you would have to take it to the IRB. And oddly enough, the IRB rejected us and said, because you're dealing with a single patient, this is clinical practice, it's not research. And we said, when does it become research? They said, once you have 3 patients, then it's generalizable knowledge. We thought, okay, all right. So we actually do have an IRB protocol that we bootstrapped over the entire process. And we beg the IRB to cover us so that we would have proper protection for human subjects. And they eventually agreed to it once we twisted the whole process into making generalizable knowledge. So technically speaking, no, you don't need to go to the IRB once you've done this. What you actually need, assuming it's, of course, an approved drug, you actually need the clinician being willing to do this. And yeah, they will have the discretion to use an approved drug in a novel way if they deem it clinically appropriate. Of course, there's a lot of risk inherent in that. And so in our experience in working with clinicians, what they want is evidence. They want to know, okay, what's the safety profile of this? What are likely side effects? And what's the evidence this could actually make a difference? So we focus on providing that in what are called research reports. Now, there's another tricky thing too. So we have a well-defined way of writing these research reports. And when I say well-defined, I mean, the lawyers at UAB were heavily involved in drafting the structure of these reports. Because some of these reports go out across state lines. They never go back to a patient, actually. They go straight to a treating physician, and they're strictly factual documents that just highlight, scientific facts known from either a data set or a paper. And it allows, the physician then to reconstruct for themselves what might be a good thing to do. And so there's a whole process for how to do, essentially how to help people while avoiding the practice of medicine. Is what it comes down to. So it's a good question with a complicated answer, but at the end of the day, that's kind of all the hoops you have to jump through to pull this off.
So a related question is the scalability of this N of 1 treatments and N of 1 data, because if you don't pull all those together or put them in the literature, you would never find them in your NLP tool. And so that's a challenge. And then how do you scale that up? As our medical system is based on, RCTs based on lots of patients, or more than 3, I guess, based by your IRB. What's your idea on scaling this then out, moving it forward?
Yeah. So in terms of, scaling this, I don't think we'll ever find a day for the patients that we see where we land on an RCT that provides us the answer. It seems so unlikely that we'd see that. So we're often left trying to tell a molecular story instead, and we draw heavily from basic science literature in most cases, it oftentimes ends with sort of optimistic speculations, as I might put it, that if you put all these facts together, it implies this might help with the sort of core molecular problem, and hopefully then that will make a difference in the patient. That's sort of how it ends right now. And then there's the question of, okay, well, how do we measure if we're making a real difference? And this, at first glance, seems almost impossible because you have to sort of come up with this process every single time. However, there's enough commonalities in rare genetic disease, at least, that you can come up with some, there's no universal endpoint, but there's some that cover a lot of different patients. So, for example, seizures are present in a very large fraction of all rare genetic disease patients. And so there's a very good chance that seizures will be a very, observable endpoint for most of the patient, or for not most, but a large fraction of the patients that reach out. So we have focused on trying to find, these, high coverage readouts that cover large swaths of rare disease space and allow us to measure the impact of an intervention, even at the level of an individual patient.
Yeah, and there's a question in the chat about, I think that's a relatively similar question. What's the acceptability of implementation for the N of 1 trials?
Yeah. So what do you mean by acceptability of the implementation in that case?
Yeah, Malabika, maybe you can
Yes.
ask.
Yes. So primarily trying to understand, like the concept of N of 1 is not as prevalent, like when it comes to clinical trials. So whenever any proposal is put forward about an N of 1 trial, how well is that accepted?
Yeah, okay. So I see what you mean. Well, as I was alluding to earlier, in some sense, the IRB will sort of step out of the way when it's N of 1. They will say, it's not our job because this isn't science. I mean, if the N is that low, they say you can't do science on 1. Which, gosh, you could definitely argue that. Because I mean, like, what do you mean by science at that point? In any case, in that sense, there's no barrier, but yeah, there is this broader issue of, you make this intervention, how do you know if things got better? That there's a real issue there. And I think what we're finding is that there's really, you have to be very creative about this. But I'll tell you one way you get around it. And so if the patient does have seizures, you can look at the difference before and after. And if the patients, or their family is willing, you could consider sort of a pulsing trial where you take them on the treatment, put them on the treatment, wash them out, put them on, wash them out, several cycles of this. In that sense, you amplify the N within a single patient, and you can pull more, you can get more confident about your findings in that case. These are hard trials, though. These are very hard because as soon as the parents see an effect, good luck. And for good reason. you're gonna have a hard time getting them off. And I'm very sympathetic to the parents that don't want to do a washout once they see an improvement. And ethically, that's probably the right thing to do. It just means that the science is hard. It's complicated by the ethics, and at the same time.
Maybe I can ask you the last question. I'm gonna make sure that your talk and message gets onto our rare disease group here. We have one in our children's hospital. We have a rare disease center. And so maybe, because I know they'll ask me this, what's the positive hit rate from the NLP database that you have? How many, you showed obviously these great stories, which are really, truly amazing. But I think there's always this skepticism of, oh yeah, that was N of 100, that one works.
Yeah.
Do you have a sense of that?
Yeah, so there's a funnel to it, and at this point, is probably 30% of all cases start with something identified. But of those, maybe a third, so 10% of all the cases originally actually go all the way to the point of a research report that goes back to a physician that also then ends up with the physician saying, I think this made a difference.
Yeah.
So, it definitely, it funnels down, but if it's 10% of everybody who reaches out, that's not so bad, given that everybody's essentially on an intractable journey to that point. And I will say it's early days, but the advent of large language models have really changed things very quickly. So we're playing with a number of them, building some of these on our own, and the hit rate has absolutely jumped in just the past few months. And so we don't know what the long-term implications are in terms of overall number of patients that it will be helped by. But it does seem like we've just hit a big bump.
Yeah, so that's fantastic. Yeah, I really appreciate the talk. I'm sure everyone will, and we will, as I said, I'll speak to our rare disease group, and hopefully they'll test out the algorithm and, make it more usable. Yeah.
Wonderful. I'm happy to engage. Just keep me posted.
Fantastic. Thanks, Matt. I appreciate it.
All right. Thank you.
Bye.
Bye.